What if one of the most powerful cancer-fighting weapons on the planet wasn't a drug, wasn't a surgery, and wasn't a chemotherapy agent, but a living organism so small you'd need a microscope to see it? What if the answer to one of humanity's most feared diseases has been sitting inside your gut, your soil, and your fermented foods all along? Here's a question that should stop you in your tracks. Why do some people develop cancer and others, exposed to the exact same toxins, the same stress, the same diet, never do? Why does one person's immune system catch and destroy a mutated cell while another person's immune system stands by and lets it grow into a tumor? The answer, according to a rapidly accelerating body of research, may lie not just in your genes, but in your bacteria. Yes, your bacteria. For over a century, scientists had a strange observation they couldn't quite explain. Some patients with serious bacterial infections would spontaneously recover from cancer. Tumors would shrink, masses would regress. It seemed impossible, but a 19th century surgeon named Dr. William B. Coley took this observation seriously enough that he began deliberately injecting patients with heat-killed bacteria. His results were so remarkable that he became known as the father of immunotherapy. But then something shocking happened. His experiments were largely shelved when radiation therapy came into fashion in the 1920s. For nearly 100 years, this knowledge sat dormant while thousands of patients underwent increasingly aggressive treatments. Today, more than 100 years later, science is finally catching up to what Coley saw with his own eyes. In February 2026, researchers at the University of Waterloo published a landmark study on genetically engineered bacteria that can literally eat tumors from the inside out. Simultaneously, NCI-funded research from Columbia University showed that a specific strain of E. coli could shrink tumors, train immune cells to remember cancer, and in 50% of treated mice with colon cancer, eliminate the disease entirely. But here's the most important thing I want you to hear today, and I need you to really listen to this. While the therapeutic applications of bacteria are still being developed in labs and clinical trials, the lifestyle choices you make right now, what you eat, how you sleep, what you expose your microbiome to, are either building an internal ecosystem that fights cancer or dismantling it. Let me start with something that will fundamentally change how you think about cancer. Picture a tumor as a city, a rapidly growing, chaotic, poorly planned city. On the outskirts, near the blood supply, you have active oxygenated cancer cells dividing and spreading. But deep in the center of the city, there's a dead zone. No roads reaching in, no oxygen, no blood supply, just dead cells and suffocating darkness. This is called the tumor's hypoxic core, and conventional medicine has a very hard time getting there. Chemotherapy drugs travel through the bloodstream, which means they reach the outer edges of the tumor. Radiation works similarly. The dead, oxygen-starved center of a large tumor is in many ways unreachable, a sanctuary for cancer cells to survive treatment and eventually re-expand. But here's where nature reveals something extraordinary. Certain bacteria, particularly obligate anaerobes, meaning bacteria that die in the presence of oxygen, find that dead core to be paradise. Ideal conditions, plenty of nutrients, no oxygen to threaten them. And so, when introduced into the body, these bacteria do something remarkable. They home in on tumors like heat-seeking missiles. Scientists at the University of Waterloo are working with a bacterium called Clostridium sporogenes, which is commonly found in soil and can only survive in environments with no oxygen. The inner core of many solid tumors represents an ideal place for this organism to grow. Once inside, the bacteria can multiply and begin breaking down and defeating cancerous tissue from the inside out. Now here's the genius part. The researchers solved one of the biggest challenges that had stalled this approach for decades: how to stop the bacteria from spreading to healthy tissue as they push outward from the tumor's core toward regions with trace amounts of oxygen. Their solution involves a natural bacterial communication system known as quorum sensing, chemical signals released by bacteria as they grow. When only a small number of bacteria are present, the signal is weak. Once the population reaches a critical size, the signal activates specific genes, in this case, an oxygen tolerance gene that allows the bacteria to survive longer as they push into the tumor's outer regions. Think of it like a military operation with a built-in safety protocol. The bacteria only power up once they've confirmed they're deep inside enemy territory. Now, this is not something you can do at home. This is laboratory medicine, and these therapies are entering clinical trials. But why does this matter to you right now? Because understanding how bacteria interact with cancer changes everything about how you think about your internal ecosystem. The bacteria living inside you, right now, today, are in constant communication with your immune cells, your gut lining, your liver, your hormones. They are either training your body to detect and destroy abnormal cells, or they are creating the inflammatory conditions that let those cells slip through. And that part, the part that happens in your gut with every meal, every night of sleep, every antibiotic you take, that is absolutely within your control. Let me take you back to 1891. A young bone surgeon named Dr. William Coley is working at New York Cancer Hospital. He's frustrated. He's watching his patients die from inoperable tumors, and conventional medicine has nothing to offer them. Then he finds something in the old medical records that makes him stop cold. A patient with a large inoperable neck tumor had contracted a severe bacterial skin infection called erysipelas, caused by Streptococcus bacteria. He was expected to die, but after surviving the infection, something impossible had happened. The tumor had vanished. Coley hunted the patient down, found him alive, completely cancer-free, 7 years after diagnosis. This was not a fluke. Dr. William B. Coley pioneered the treatment of his patients with both live bacteria and a mixture of heat-killed bacteria known as Coley's toxins. He treated hundreds of patients over his career with what he called his bacterial toxins, a mixture of heat-killed Streptococcus pyogenes and Serratia marcescens. He reported impressive results in inoperable cases: bone tumors, soft tissue sarcomas, lymphomas. Some patients experienced complete remissions that lasted decades. So why did the world abandon this approach? The answer is complicated. Part politics, part profit, part the intoxicating promise of radiation therapy, which arrived in the 1920s and seemed cleaner, more controllable, more scientific. Coley's toxins were messy. They caused high fevers. They were hard to standardize. The medical establishment turned away. But here's what Coley had actually discovered. A century before the scientific language existed to describe it, he had found a way to trigger a massive immune response inside the body, one so powerful that the immune system finally awakened from its cancer-induced slumber, turned its full force on the tumor. Here's what's really going on inside your body when bacteria invade a tumor. The immune system is not passive. It is always scanning, always patrolling. But cancer is clever. Tumors evolved to suppress immune activity in their immediate environment, essentially putting up a do not disturb sign for your T cells and NK cells, your natural killer cells. When bacteria arrive in the tumor microenvironment, they trigger something called a pattern recognition response. Your immune system's sensors detect bacterial signatures, flagellin proteins, lipopolysaccharides, and it sounds the alarm. Massive inflammatory signaling. Immune cells flood in, and in that flood, they encounter the cancer cells they've been unable to see, and they destroy them. Researchers at Northeastern University, publishing in Nature Biotechnology, used non-pathogenic E. coli engineered with proteins on its surface to elicit an immune response and attack cancer cells by activating T cells and NK cells. The study found that 50% of mice with colon cancer were cured by the optimized bacteria. But here's where it gets even more incredible. When the cured mice were given another tumor, their immune system went into action and attacked, suggesting that the immune system learned to remember the cancer cells, making it harder for cancer to return. Let that sink in. Not just tumor shrinkage, immune memory, the body learning how to fight its own cancer. That is the future of cancer medicine, and it started with Coley's observation in 1891. The lesson for you today: your immune system is not broken. It may be suppressed by chronic inflammation, by poor sleep, by insulin resistance, by a disrupted gut microbiome. The question is, what are you doing daily to unsuppress it? Most people think about gut bacteria in terms of bloating, digestion, or maybe the occasional probiotic yogurt. But what the research is increasingly showing is far more profound. Your gut microbiome is one of the primary regulators of cancer risk in your body. Let me explain this with a simple analogy. Imagine your immune system as an army, highly trained, equipped with sophisticated weapons. But like any army, it needs intelligence. It needs to know who the enemy is, where they're hiding, and when to strike. Your gut microbiome is the intelligence agency. It is constantly collecting information, processing it, and feeding it to your immune cells. A diverse, healthy microbiome sends high-quality intelligence. A depleted, dysbiotic microbiome sends noise, or worse, it sends false signals that cause the immune army to stand down when it should be fighting. The gut microbiota plays an important role in tumor pathogenesis by regulating host metabolism and immune responses, and more studies are beginning to explore the potential of gut microbiota and its metabolites as cancer biomarkers. Think about what regulating host metabolism means. Your microbiome influences how you process carbohydrates, fats, and proteins. It affects insulin sensitivity. It regulates inflammation. It modulates cortisol. It interacts with your liver, one of the most important detoxification and cancer defense organs in your body. Every single one of these systems, when disrupted, creates conditions that favor cancer growth. Here's the metabolic context you need to understand. When you eat a high-fat, low-fiber, ultra-processed diet, you feed the bacteria that produce toxic secondary bile acids. These bile acids damage the lining of the colon and have been directly linked to colorectal cancer. When you eat a fiber-rich, plant-forward diet, you feed bacteria like lactobacillus and bifidobacterium, which compete with the harmful strains and shift your gut chemistry toward protection. And it's not just colorectal cancer. Specific bacterial genera, such as lactobacillus, bifidobacterium, and akkermansia, were linked to favorable outcomes in gastric cancer, while others, like Streptococcus and Escherichia shigella, were associated with poorer prognosis. There's also something critical happening at the interface of your gut and your liver, what researchers call the gut-liver axis. When your gut lining is healthy, it acts as a selective barrier. Nutrients get in, toxins stay out. But when the gut lining becomes leaky, as happens with chronic stress, alcohol, ultra-processed food, and antibiotic overuse, bacterial toxins seep into the bloodstream, travel to the liver, and create systemic inflammation. That low-grade chronic inflammation is one of the most powerful drivers of cancer initiation and progression we know of. If this is resonating with you, consider subscribing to this channel. Every day, I break down the science of how to protect your metabolic health, because when your metabolism is healthy, your body's defense systems work. That connection matters more than most people realize. Now here's one of the most elegant stories in all of cancer biology, and it involves a molecule most people have never heard of. It's called butyrate, and it is produced by your gut bacteria when they ferment dietary fiber. Let me walk you through this. You eat a meal rich in fiber, let's say a bowl of lentils, some roasted vegetables, maybe a handful of walnuts. Your small intestine can't digest that fiber. It passes intact into your large intestine, where bacteria, particularly species like Faecalibacterium prausnitzii, Roseburia, and Eubacterium rectale, begin to ferment it. The end products of that fermentation are called short-chain fatty acids. The most important of these, from a cancer prevention standpoint, is butyrate. Now here is where it gets remarkable. Butyric acid can contribute to the prevention of colorectal cancer because it improves the intestinal barrier through the increase in mucus production and the proliferation of healthy cells. But that's just the start. Butyrate does something even more extraordinary when it encounters a cancer cell. Normal cells use butyrate as a primary fuel. They happily burn it for energy. Cancer cells, however, have reprogrammed their metabolism. They prefer glucose. This is known as the Warburg effect. When butyrate builds up in the colon at high concentrations, cancer cells can't burn it efficiently. Instead, it accumulates in the cell nucleus and begins acting as a histone deacetylase inhibitor, essentially an epigenetic switch that reactivates genes telling the cancer cell to stop dividing and undergo apoptosis, self-destruction. Think of it this way. Healthy cells eat the butyrate and thrive. Cancer cells are poisoned by the same molecule. So what does this mean practically? It means the fiber on your plate is not just about digestive comfort. Every gram of fermentable fiber you eat is a substrate for butyrate production. Every time you choose a processed food over a whole food, you're starving the bacteria that make this anti-cancer compound. The research is also showing that butyrate plays a role beyond the colon. It helps regulate immune cell development. Specifically, it promotes the production of regulatory T cells that prevent excessive inflammation. It supports the blood-brain barrier. It influences insulin sensitivity. The more we look at butyrate, the more central it appears to total body health. So here's your butyrate boosting protocol. Start by adding at least 25 to 35 grams of fiber per day. The best sources for butyrate production are resistant starches, slightly cooled rice, cooked and cooled potatoes, green bananas, legumes. Also crucial are fermentable fibers from garlic, onions, leeks, asparagus, and chicory root, which contains inulin. Pair these with regular consumption of fermented foods, kefir, kimchi, sauerkraut, natural yogurt, to ensure you have the microbial workforce capable of doing the job. One more thing to know, you cannot supplement butyrate as a pill and expect the same results. The magic isn't in taking butyrate, it's in growing the microbes that produce it continuously in your own colon based on what you feed them. We've established the mechanism. Now let's get precise about the strategy. Your microbiome is not fixed. It is one of the most dynamically changeable systems in your body. Studies have shown measurable shifts in microbial composition within as little as 24 to 48 hours of dietary change. This means that the choices you make today, at your very next meal, begin shifting the balance of your internal ecosystem. Let me explain this with an analogy. Think of your gut as a garden. Different plants thrive in different soil conditions. If you pour pesticides on your garden every day, antibiotics, alcohol, ultra-processed food, you kill the sensitive, beneficial plants first. The weeds, the resilient inflammatory species, survive and take over. The garden looks full, but it's full of the wrong things. And those wrong things produce the metabolic toxins and inflammatory signals that increase cancer risk. Now, what does the research say about specific foods? Gut bacteria break down fiber and other compounds from fruits, vegetables, grains, legumes, nuts, and seeds, producing metabolites with various health benefits. Certain plant foods also stimulate the growth and activity of specific gut bacteria, leading them to produce large amounts of anti-inflammatory and anti-cancer compounds. Particularly exciting is research on a class of compounds called L-glutanins, found in walnuts, berries, and pomegranates. Gut bacteria convert these into compounds called urolithins, which have demonstrated powerful anti-inflammatory effects on colon tissue and are being studied specifically for colorectal cancer prevention. Gut microbiome compositions associated with high fiber intake, characterized by high diversity with high relative abundance of Ruminococcaceae, butyrogen-producing species, and methanogens, are associated with better immune responses, including improved response to cancer immunotherapy. This is enormous. It means that people who eat high-fiber diets don't just have healthier digestion. They have immune systems that are better equipped to fight cancer and that respond better to cancer treatment if it becomes necessary. The foods you want to build your microbiome around. Fermented foods daily. Kefir, yogurt with live cultures, kimchi, sauerkraut, miso, tempeh. Prebiotic-rich vegetables, garlic, onions, leeks, asparagus, Jerusalem artichokes. Polyphenol-rich foods, berries, dark chocolate at 85% or higher, green tea, pomegranate, extra virgin olive oil, resistant starches, cooled cooked rice, lentils, chickpeas, green bananas, omega-3 rich foods, wild salmon, sardines, flax seeds, walnuts. These reduce the inflammatory tone of the gut. The foods that actively harm your microbiome and raise cancer risk. Ultra-processed foods with emulsifiers like polysorbate 80 and carboxymethylcellulose. Studies show these directly damage the gut mucosa and promote dysbiosis. Refined sugar and high fructose corn syrup. Feed inflammatory bacterial species and spike insulin, which promotes cellular proliferation. Excess alcohol disrupts the gut lining and shifts microbial balance toward harmful species. Overuse of antibiotics without necessity. the single most dramatic disruptor of microbiome diversity. Most people understand that food affects the gut. Fewer people understand that your nervous system, your hormones, and your sleep quality are equally powerful forces shaping your microbial ecosystem and therefore your cancer risk. Here's the hormonal picture you need to understand. When you are chronically stressed, your body releases elevated cortisol. Cortisol, in short bursts, is protective. It mobilizes energy and prepares you for a threat. But cortisol chronically elevated day after day does several things that are deeply relevant to cancer biology. First, it suppresses the activity of natural killer cells, your NK cells, the immune system's frontline cancer patrol. NK cells are designed to detect abnormal cells and destroy them before they form tumors. Chronic cortisol reduces NK cell activity significantly. Second, it increases intestinal permeability, the leaky gut we discussed earlier, allowing bacterial endotoxins to reach the bloodstream and create systemic inflammation. Third, it disrupts the gut microbiome directly. Studies show that chronic psychological stress alters microbial composition, reducing the populations of beneficial lactobacillus and bifidobacterium species. Sleep is the body's primary cortisol reset mechanism. During deep sleep, particularly slow-wave and REM sleep, the body restores immune function, clears inflammatory compounds, and resets the hypothalamic pituitary adrenal axis. Consistently sleeping less than 6.5 hours per night has been associated in large population studies with reduced NK cell activity and increased cancer risk. The gut-brain axis is also bidirectional. Your microbiome produces over 90% of the body's serotonin, as well as GABA, dopamine precursors, and other neurotransmitters. A disrupted gut sends stress signals to the brain. A stressed brain disrupts the gut. This is a reinforcing cycle, and breaking it requires intentional, consistent lifestyle inputs. Your stress cortisol microbiome protocol targets 7 to 9 hours of sleep nightly in a dark, cool room between 65 and 68 degrees Fahrenheit. Use morning light exposure within 30 minutes of waking. This anchors your circadian rhythm, which directly governs cortisol pulsing and immune cell activity. Practice 10 to 15 minutes of daily breath work or meditation. Even simple box breathing, 4 counts in, 4 hold, 4 out, 4 hold, has been shown in clinical studies to measurably reduce cortisol. Limit blue light after sunset to protect melatonin production. Melatonin is not just a sleep hormone, it is a potent antioxidant with direct anti-tumor properties. Consider adaptogens, ashwagandha, and rhodiola. Rosea have evidence for reducing cortisol response to chronic stress. Exercise regularly, but not excessively. 30 to 45 minutes of moderate intensity movement most days is strongly associated with microbiome diversity and cancer risk reduction. Overtraining has the opposite effect, elevating cortisol and suppressing immunity. One more key point. The relationship between insulin, glucose, and cancer is not separate from the microbiome conversation. It is deeply intertwined. High insulin levels, driven by a high carbohydrate, low fiber diet, directly promote cellular proliferation and suppress apoptosis. A healthy microbiome improves insulin sensitivity, partly through butyrate production and partly through regulating bile acid metabolism. When you fix the gut, you often fix the metabolism. Let me step back and give you the full scientific picture of where bacteria-based cancer medicine is right now, because it's moving faster than most people realize. We are no longer in the era of theory. We are in the era of clinical translation. Bioengineers have armed bacteria with stronger tumor sensing and more targeted drug delivery capabilities and improved control of off-target toxicities. An increasing number of therapeutic bacterial strains are now entering clinical testing, promising to enhance the efficacy of conventional anti-cancer treatments. Here's what's being studied. Salmonella typhimurium has been extensively modified to be safe in humans while retaining its natural ability to invade tumors. It can be engineered to deliver cytotoxic compounds directly into cancer cells or to activate the immune system inside the tumor microenvironment. Multiple clinical trials are underway. E. Coley 1917, a probiotic strain that has been used for more than a century, has been engineered in NCI-funded research to deliver interferon gamma, a protein immune cells rely on to detect and attack cancer directly to tumors. Combining this with an immunotherapy drug kept tumor growth in check longer than either treatment alone. Listeria-based therapies for pancreatic cancer are particularly exciting because pancreatic cancer is notoriously difficult to treat. It's often called the silent killer because it metastasizes before the primary tumor is even diagnosed. Radioactive listeria injected intravenously into highly metastatic pancreatic tumor-bearing mice could deliver radioactivity specifically to metastases without harming normal tissues, because the immune system efficiently clears bacteria in normal tissues while the immunosuppressed tumor microenvironment allows them to accumulate and persist. And now there's a new frontier, theranostics, using bacteria simultaneously to detect, image, and treat tumors. By engineering bacteria to produce light-emitting proteins alongside therapeutic payloads, scientists can track exactly where the bacteria are going inside the body and confirm they've reached the tumor, all while delivering the treatment. What does all this mean for you today? It means this field is real. The science is solid, growing, and moving fast. It also means that the microbiome you build now, through diet, sleep, stress management, and avoiding microbiome-damaging medications when unnecessary, may not just affect your health today. It may affect how your body responds to these next-generation therapies if you ever need them. Research is already showing that patients who go into cancer treatment with a healthy, diverse microbiome respond significantly better to immunotherapy than patients with depleted microbiomes. Your gut bacteria help determine whether the drugs work. This is not a footnote. It is a central finding in modern oncology. Let me now bring everything together into a clear daily protocol you can start implementing immediately. This is not a cure. This is a lifestyle architecture designed around the science we've just discussed. Optimizing your internal microbial ecosystem to reduce inflammatory cancer-driving signals, maximize immune surveillance, and create a gut environment that works for your health rather than against it. Morning, wake at consistent time. Your circadian rhythm governs immune function. Get outside for natural light within 30 minutes of waking. Hydrate with water before coffee. 500 milliliters of filtered water, ideally with a pinch of mineral-rich sea salt and a squeeze of lemon. This supports liver bile production and gut motility. First meal, incorporate at least one prebiotic food and one probiotic source. Example, Greek yogurt with berries, walnuts, and a tablespoon of ground flaxseed. This gives you probiotics from the yogurt, L-gitannins from the berries and walnuts, butyrate precursors from the flax, and fiber. Midday, build your largest meal around vegetables and legumes. A simple target, half your plate should be non-starchy vegetables. Include a palm-sized portion of protein, ideally from wild fish, eggs, or legumes. Add fermented foods as a condiment, kimchi alongside a grain bowl, miso soup, or a small amount of sauerkraut with your main dish. Avoid liquid sugar at this meal. Fruit juices, sweetened drinks, these rapidly shift your gut microbiome toward inflammatory species within hours. Evening, finish eating 3 hours before bed to allow your gut to enter its overnight fasting repair state. A small cup of green tea is fine. EGCG is a potent polyphenol that supports beneficial microbes. Prioritize sleep, dim your lights, avoid screens 1 hour before bed, room temperature at 65 to 68 degrees Fahrenheit. This is when your immune system consolidates, when NK cells recharge, and when your gut lining repairs. Weekly additions, 3 to 4 times per week, 30 to 45 minutes of moderate aerobic exercise. Studies show this is one of the strongest non-dietary drivers of microbiome diversity. Once per week, intentional stress reduction practice, a long walk in nature, a sauna session, heat shock proteins, support immune function, or a longer meditation practice. Monthly, evaluate your antibiotic history and avoid unnecessary use whenever medically appropriate. Every antibiotic course can reduce microbiome diversity for months. Supplement considerations. A high-quality, multi-strain probiotic with 5 to 10 strains and 10 to 50 billion CFU taken with food can help maintain diversity, especially after antibiotic use or during periods of high stress. Prebiotic supplements, inulin, FOS, or resistant starch powder can amplify butyrate production when dietary fiber is insufficient. Vitamin D3 plus K2, vitamin D profoundly supports immune function and has independent anti-tumor signaling properties. Research consistently links low vitamin D with increased cancer risk. Target serum levels of 50 to 80 nanograms per milliliter. Omega-3, EPA plus DHA, 2 to 3 grams daily. Powerful anti-inflammatory, supports gut lining integrity, modulates NK cell activity. Let me bring this full circle. Over the last 130 years, humanity has been searching for a silver bullet for cancer. We've developed surgery, radiation, chemotherapy, targeted therapy, and immunotherapy. Each advance has saved lives. But we have largely overlooked one of the most ancient and powerful allies we have, the microscopic world inside and around us. What the science is now making undeniable is this: bacteria are not just pathogens. They are not just good guys and bad guys living in your gut. They are active participants in your immune function, your metabolic health, your hormonal balance, and, increasingly, we understand, your cancer defense system. Here's what you learned today. Specific bacteria can home in on the oxygen-starved core of tumors, a place no drug can easily reach, and destroy cancer cells from the inside out. This is not science fiction. It's in clinical trials. William Coley showed us a century ago that triggering an immune response with bacteria could cure incurable cancers. Modern immunology is explaining exactly why that worked and building on it rapidly. Your gut microbiome is not a passive bystander. It is a cancer defense system. Its diversity, its activity, and its balance are shaped every day by what you eat, how you sleep, how you manage stress, and what medications you take. Butyrate, produced by your gut bacteria when they ferment fiber, signals cancer cells to self-destruct while feeding healthy cells. You build butyrate by feeding fiber to the right bacteria. Diet is the most powerful lever you have over your microbiome, and your microbiome is one of the most powerful levers you have over your cancer risk. Sleep and stress management are not optional wellness habits. They directly govern NK cell activity, cortisol balance, gut barrier integrity, and the anti-cancer capacity of your immune system. Here's your direct challenge. Try just one change from this video today. Add a fermented food to your next meal. Go outside for sunlight first thing tomorrow morning. Replace your afternoon processed snack with a handful of walnuts and berries. Track how your body responds over the next 2 weeks. You do not need to overhaul everything overnight, but every single choice, every meal, every night of good sleep, it compounds. Your microbiome is being shaped right now, in this moment, by your last meal and your last night of rest. The question is whether you're shaping it consciously or letting it drift. Cancer is not inevitable. Inflammation is not inevitable. A depleted, dysbiotic gut is not inevitable. You have more control over your biological terrain than most doctors will ever tell you, and the science is on your side. If this video added value to your understanding of how your body works, please like it, share it with someone who needs to hear it, and subscribe to join a community of people who are serious about optimizing their health naturally, without gimmicks, without hype, just science and strategy. I'll see you in the next video.